Okay. Hello, ladies and gentlemen. Good morning or good afternoon to everyone. This is Emily, the BD Director at NanoBiosensors. Welcome you to join this webinar, a new technology for rapid determination of GFAP. So today, I'm pleased to invite our CEO, Tony, and our Dr. Zhou from Zhongzhen University as our speaker of this webinar. And during this meeting, welcome to leave the message to chat box or Q&A part to me if you have any questions, and we will answer all of it later. So during the webinar, we have three major sections. The first one is introduced by our CEO, Tony. He will introduce our FOPP technology and also introduce what analyze or function that FOPP technology could offer. And the second one is introduced by Dr. Zhou. He will introduce the research which he is doing now and how he do discover the details through FOPP technology. And the third part is Q&A section. So let me introduce Tony and pass the time to Tony. Hello, Tony.
Thanks. Yeah, thanks, Emily, and good to see all of you through our Instant NanoBiosensors webinar. So today, we would like to talk about more deeply about our ultra sensitivity, and we welcome Professor Zhou also mention about how we approach this outstanding performance. Regarding the Instant NanoBiosensors, we believe the light can save lives. And we would like to become the pioneer and leader of biomarker solution provider from the analysis to diagnostic. Before we introduce to our product and technology, I would like to just some update news for you to know. In 2020, we'll be selected from America as a strategic partner. So as you can see on the website, their Merck Innovation Center that mentioned about our company. And during these past 2 years that we have co-hosted 3 global webinar with the UK, which is the biggest antibody company globally, and talk about our product and technology. So later I will introduce more about these details. Normally, we talk about when we get sick, so we go to the hospital because the hospital, their commission, like a central lab or hospital has the invasive or some capital equipment. So, but normally, just like an iceberg, only can see the upper of the iceberg. So we use the existing solution to estimate how deep and how widely of the iceberg. So according to a reference, there's a 75 human protein are difficult to detect because of the low abundance. So regarding this, as you can see, through our licensing biomarker analyzer, we can deeply and widely to detect the more sensitive than the traditional methods. So this become the can find the centralization to decentralization. Just about we are the first in the world to use the licensing, which is combine optical fiber, nanotechnology, and microfluidic. And the strategy is from the life science analysis to diagnostic detection. We have over 40 patents globally. So and in 2021, now we have 4 to 5 more publications. And that's we describe a deep our core technology. Our core technology, which divide into 2 parts. One is the FOPP, which is a fiber optical particle plasma resonance. So use optical as a sensor to combine with antibody or antigen that we can sense the difference between in serum plasma, or we can do how the affinity, those kind of things. And the other things, we have a global patents called the in-chip, which is we don't put any, you don't need any pumping tubing or any of the rest. So it's an auto folding chip. So combine the optical sensor and put it into our in-chip auto folding chip, then can become the our core technology, can provide the high sensitivity down to nanogram per milliliter and only need a couple steps. So some information just I need to know. As you can see, other instrument capital equipment that would like to approach the same performance that need a very complicated optical design. So the inside, as we know, the laser or the detection module, and also the fully modules, for example, like pumping, tubing sinks, and also the rest of the liquid anode is quite complicated. So because we are in the field around like 20 years, so we know for our in-chip technology that can reduce a lot of the after-service loading. So we combine these two technologies together. So we do a very different go-to-market strategy is from the analysis to the detection. So for the life science of the analysis, the scientists, they focus on the antibody drug, antibody selection, or vaccine development, and also the ELISA biomarker assays. That's a quite regular experiment in the laboratory. And then apply the use the same machine and technology, use the detection, can do the diagnostic, monitoring, and lots of different applications. So to describe the mode analysis methodology, for the antibody antigen selection, we know the ability analysis like K-on, K-off, and KD ability. And also use our epitome binning to recognize the different epitome and can be as like an ELISA pair. And use our analysis platform that can do the buffer selection to find out which the buffer is the best of the stability. And also we talk about the performance analysis like limiting of detection and linear diameter range, this kind of part. So for the first of the analysis part, we can not only do the analysis, but also use one antibody to do the detection. So most of the later I will use some example just let you to know. Some of our clients that use the deeply analysis to help their ELISA kits or their fast rapid test develop. Yeah. So first part is the biomarker analysis. Use our licensing biomarker analyzer. So why we need a FOPP analysis before immunoassays? As you can see, that there's a very complicated and lot of the possible can make us our ELISA or antibody develop become a trouble, right? So when we go to the some customer side and discuss with the scientists, they always feedback why my biomarker assay is always the West out time has a big specificity, sensitivity, and the linear diameter range is not that good. So after they running through those of the clinical sample, they are not sure about their ability, those kind of things. Yeah. So use a little back to our laboratory work. I think we all familiar with one of the things. When we want to run the good like a nucleic acid analysis apply on the like real-time PCR or NGS, we will definitely use like a nail drop, qubit, or biomarker analyzer to do the quality control. But it's very funny that during this moment, we use a protein analysis is by the ELISA, and the detection is also the ELISA. So how come we do use the ELISA as an analysis and also use the ELISA as a detection? So our solution is to provide the deeply analysis before we apply to the next step of immunoassays, which can provide a good result, cost save, and also the time saving. How we run this experiment is quite easy. We just pick up our influence chip Inside the input, our input chip, there's a light sensing. It's a 4% sensor there. So we use an antibody. When we immobilize that, and through the immobilization, our real-time curve, you can see how antibody immobilized good or not before we go to the next step. If we know the immobilization of the antibody is good, then we can go to the second step of the analysis detection. Yeah. So as you can see, if the antibody and analytes can have a very good affinity and also the binding is good, then you can see the reduction curve. But if it is not, then you will see the no any response. So based on this, that we can do the lots of the different possibility. Later I will introduce. One of these we do is the affinity assay to help the scientists to find out the protein-protein interaction. For example, like association and dissociation, these kind of the experiments. So based on the what we mentioned about the in-curve, these kind of the assays, they can provide the scientists like not only the affinity, not only the epitope binding, but also you can use our system to do the buffer selection to find out the best of a combination of the protein-protein interaction and also the environment, those of the buffer scenes. And also to find out one of the issues is the performance of the analysis. Later I will show you why the Abcam US, the biggest antibody company, they would invite 3 times give a speech because we use their antibody but provide the outstanding performance onto our platform. Yeah. Just back to a little bit about the scientist part. What is the affinity ability? Affinity ability is how fast does these two antibody or antigen associate, and the dissociation is how fast the compounds fall apart. So we based on this, we will help scientists to find if the buffer and the interaction is good. So they will locate on the class 1. They will be the perfect match. So based on this, then we have what I mentioned why we invite by the Abcam, because we use a very experiment to help the scientists understand our performance. We use the R&D system. They use their kit, which is combined antibody and antigen and also their buffer. But we replace one of the Abcam antibody as another experiment. So as you can see, they have a different antibody combination and also the affinity. But it's very interesting is our R&D system, they use their like a commercial kit, but they actually, because the buffer is different, so the location is on the class 2. But the Abcam that we use the PBS and also the R&D system antigen and also the antibody and locate on the class 1. And actually the good combination is Abcam antibody and also the R&D analog. So we also always, use in our database to compare. It's all located on the below of the other section of the affinity. So which means the Abcam of these combinations is the best of the choice. The other example for you to know is epitope binding. For our part is we can use one chip and simultaneous to look into 4 different kind of the epitope. Just for you to know one of the example, which is we already have a publication and already one of the biotech company use our epitope binding to develop the V strip of the COVID-19 antigen rapid test. So based on these experiments, they have many different antigen. So that use our epitope binding to find out the best of the match of the pair. As you know, like ELISA or fast rapid test always need 2 antibody. So use our epitope binding can find out the best pair of the these combination. That's the first. And secondly, due to the anti-immune this company, they are working on the antibody drug. So they also use these antibody binding to focus on how to develop a new binding epitope for the antibody developed. And the other experiment, just letting you to know, because our company is focused on the neuron size part. So we have a biggest clinical trial is around like 1,000 samples. So how we run this experiment? First, we select a lot of the different combination and also the commercial antibody from the different companies. So first we screen, and secondly we just use our epitope binding to find out the best of match before we have NFL these commercial kit. So based on these analysis, we will have and also looking for the neuron size cooperation to understand more deeply of the brain biomarkers. And then some of the clients they might ask how kind of sample that can apply to our analysis. There's many different possibilities, and also we have over 30 publications, all based on the different combination of these, like antibody, antigen, receptor ligand, DNA or protein or DNA, and DNA, those kind of analysis for these kind of the analysis. So the second section will mention about the readers about the detection. I will go a little faster to get you to know how we run through these biomarker detection. As we know, to do the detection, we have two very unique part. One is ultra sensitivity, can down to the femtogram per milliliter, and the other is for the wild linear range up to 5 orders. Some of the scientists might ask, or some of the clients might ask, why we need these ultra sensitivity? Because it's for the prediction or early phase disease and non-invasive, those kind of the detection. Also to do the health management, these kind of the benefits. And for the wild linear range, it helps the scientists and help the clinician to do from the central lab to the on-site detection. And one of the clinicians that mentioned about the immune response, they are not only looking how deep, how sensitive they would like to achieve, but also they would like to see the immune response and to reduce the drug treatments. Yeah, so regarding our centralization and also the differentiation, we know that some of the company in Europe or in US, they have a different kind of the methodology. But we use our licensing biomarker analyzer can do only loading detection to achieve the outstanding performance. One of the very famous company is Quanteness. Their CEO in 2021, JP Morgan, is mentioned about, during this pandemic, we are looking for a decentralization solution because they already have a centralization Quanteness machine. So they are looking for a desktop, but also keep the simple and easy handling, can help the scientists to do the ultra sensitivity and wild linear range, these kind of a brain biomarker detection. So we have a different detection. Today we will cover two. One is a direct assay. The other is a ELISA. I think later Professor Zhou will mention the more information about the ELISA. So I will use the direct assay as an example for you to know. Based on these, we already have several publications. We welcome you browse through our website to have more ideas. And also we have some scientists and working on the clinical cooperation. So based the direct detection assay, we can use only one antibody. You can use your antibody immobilized by yourself and to detect a nanogram and picogram, those kind of labels, and with a label-free, these kind of features. So one of the examples that just saying you to know, we use a TNF alpha, and the sample actually is from the yeast flu. So the yeast flu, the inside would detect how the TNF alpha and MMP3, these kind of cytokine their concentrations. So as you can see, the red line that means mention about the ELISA, traditional ELISA curve. Our FOPP analysis can provide a deeper sensitivity and also wide linear range up to 5. And also the other MMP3, this biomarker, has a narrow of the data. We not only provide the wide linear range, but also have the ultra sensitivity. Yeah, and based on this, and
Hello, Tony. I think you are mute. Please open your mic. Yeah.
Based on this, then we have
Yeah,
for ELISA assay, just let you to know is
with the different methodology. And for based on that, use a sandwich assay, just like a two antibody to approach this kind of outstanding performance. So this kind of a paired combination that can down to pentagram per milliliter and also provide high specificity and high ultra sensitivity and wide linear range. We already have an impact factor. Actually, this publication is up to 10.2 impact factor. So it's used as sepsis, this kind of the data to help the scientists to do the develop. I think later Professor Zhou will mention more about that. And then these are all our product portfolio to let you to know. Now we have a MB D200 is 2 channels. And this year we will have 8 channels. And next year we will have an Auto D16 to do the fully automatic liquid handling device. And they're all based on the same chips. So work on these projects quite easy. We've been have a lot of experience with scientists. So once the candidate validation and do the data analysis and go to the next step for the detection and analysis part. Now we have a couple projects running. So just information for you to know. So this is all our team, excellent, outstanding team, just letting you to know. Later, I will pass to the Professor Zhou to have a more deeply analysis and sharing. Yeah, thank you.
Thank you, Tony. And thank you for your sharing and introduce the analysis ability of FOPP platform. It is not only affinity analysis, but also aptal binning or even the performance analysis such as LDR or LOD, or even the novel protein quantification. So then let us welcome Dr. Zhou to introduce his research. And let me pass the time to Dr. Zhou. And Dr. Zhou, please open your mic. Yeah, thank you.
Hi, ladies and gentlemen, and here I would like to share with you about how I use the FOPPR technology for rapid determination of GFAP. Okay, as you know, the stroke is the second leading cause of death globally, and each year there will be about 30 million of people having stroke, and then about 40% would die each year, and 15 to 30% suffer from permanent disability and causes over 100 billion US dollar per year. So that's a very important issue for the health and to take care to develop more sensitive assays. And there are 2 types of strokes. One is the intracerebral hemorrhage, ICH, and another one is ischemic stroke. And for the ICH, there's do not have any drugs available for treatment. However, for the IS, there's some drugs available for treatment. So that's very important that we can differentiate the two kinds of stroke after the onset of the symptoms. And one of the very important biomarker today is a GFAP. It's an intermediate filament protein found in astrocytes in the central nervous system. And following traumatic brain injury and stroke, actually the GFAP and its breakdown product can rapidly release into the biofluids, including the CSF and the blood. But they are following different temporal profiles. For example, the ICH, they release in less than 4 hours, but for IS, it's over 24 hours. That's why we can make use of this kind of time window probably to differentiate the ICH and IS. And there's some literature actually reporting that actually after the onset of stroke, actually the GFAP concentration in serum actually is much higher for ICH than IS. That's as a result, actually, there's a so-called golden hour about after the onset of 6 hours to differentiate between the ICH and the IS. As you can see, the ICH actually would happen, would release to the blood much earlier, but the IS, the GFAP will be released to the blood maybe after 24 hours. So if we can detect the GFAP within the 6 hours after the onset, that would be very critical to save the life. Here, I would like to introduce the technology, the FOPPR technology. How can we do that? Actually, we have optical fiber, and then we put the gold nanoparticle on the fiber core surface, and then we conjugate the receptor on the surface of the gold nanoparticle, and then we shine a green light beam from the LED to let it propagate along the optical fiber. So during the propagation, there will be a Watson wave outside the fiber core to interact with the medium outside. And then this interaction actually will be amplified by the so-called total multiple internal reflection. So that means if there's some golden particle at the core surface, they will absorb the green light, and then the signal at the end of the fiber would decreases. And this signal will be enhanced by the length of the optical fiber of the sensing region. And suppose if we have an antigen would bind with the antibody, actually the absorbance of the absorptivity of the gold nanoparticle will increase. And this kind of increase of the absorbance would generate a real-time sensogram that we can know how much would be the binding happen. So this kind of so we call it direct assay is a label-free. We don't need to label the molecule, the analyte, and also we give a real-time detection. And the detection limit is quite good. Actually, can force into a picomolar to nanomolar. However, for some critical application, for example, for the liver diseases, this kind of detection limit is still not enough. So recently, we developed a so-called new technology called the fiber optic nano-gold length sorbent assay. We call it FONISER. Actually, there's a kind of FONISER using the same instrument, but we just make a different chemical arrangement. Here now we instead of putting the gold nanoparticle on the core surface, we put the detection capture probe on the fiber core surface, and then we put a detection probe conjugate with 3 gold nanoparticle, and then after the interaction, the analyte would interact with the 2 probes to form a sandwich assay, and then that would make a very high sensitivity. The reason is very simple, because for the direct assay, the change of absorptivity is much smaller as compared to the FONISER that original don't have any gold nanoparticle in the surface, but after the sandwich, they would have gold nanoparticle on the surface, and the change would be in proportional number of gold nanoparticle on the surface. That means the alpha is much greater than the delta alpha, and then that causes the sensitivity increased by several orders, and the detection limit falls to sub-femtomolar to picomolar. And here is the sensorgram, real-time sensorgram for the constructing the calibration curve of the GFAP from 0.01 picomole gram per ml to 1 nanogram per ml. And then there's a calibration curve. The detection limit actually can down to 57 attomolar. And that's I think that's a very amazing detection limit. And the linear dynamic range have 5 orders. I think that's very important, useful for the diagnostics. And before we go into real sample, we want to take some interference tests using the serum. So we fry some standard samples into the serum, and then dilute it to 20 times. And then actually from the recovery result, it's very good. Actually, it's just lot more than 10%, and the CV value actually is smaller than 4%. And as you can see, actually, the spike sample fall in the same calibration curve. And to validate our method, we compare with the traditional ELISA method. As you can see, we have a much lower detection limit and wider linear dynamic range as compared to ELISA. And the LOD, the detection limit, at least 4 order of magnitude lower than the ELISA, and the linear dynamic range 5 order as compared to 2 with the ELISA. And we also want to validate it with some real samples. That the real samples provided by Dr. Tang from National Taiwan University Hospital. And when we receive the sample after collection, centrifuge for 20 minutes, and then we add some PBF buffer and then store it at -20 °C until used. And here's the result from our analysis of real samples using both the FONISER and the ELISA for the GFAP. As you can see, actually, the comparison, the results are quite good. You can see that the ELISA result and our FOPP result have highly correlated. Actually, the slope is about 1. That means the 2 methods don't have any bias. And we have about 13 real samples, and 10 are positive from clinical viewpoint, and 3 elective from the clinical viewpoint. And for our analyzer, we all detect it correctly. But for the ELISA, there's a one false positive. So as a result, the diagnostic specificity is 100% for FOPPL, but 75% for ELISA. And the positive predictive value is 100% for FOPPL, but 90% for ELISA. And the test efficiency is also 100% for FOPPL, but 92% for ELISA. So that demonstrates that our method actually is very useful for clinical sample. So now I would like to compare our technology with some very sensitive technology, including the traditional ELISA method. And one of the very sensitive methods now is the Simoa, that's by the Potteries company. We compare their two kinds of instruments. And another very popular method is electrochemiluminescence immunoassay. And from this table, you can see that our LOQ actually is significantly better than the Simoa, if that considered to be a very high sensitivity method. And our linear dynamic range also is the best compared with other three methods, the 2, 3, and 4 order, but we have 5 order. And the running time also is very, very short, it's about 15 minutes, but for other methods, over hours. So we have very high sensitivity and rapid time. And I think that demonstrates that our FOPPL technology for ELISA method is very useful, maybe possible in the future for a lot of neurological diseases. Thank you for your attention.
Thank you. Thank you. Thanks a lot for Dr. Zhou sharing your research with us and showing the great result with FOPP technology and FOPP platform. And so let us get into the third section, Q&A section. I got a few questions here, and I will pick 2. One is for Dr. Zhou, and the other is for Tony. So the first one is for Dr. Zhou. So Dr. Zhou, the question is, could you mention more detail about ELISA assay of FOPP platform? And what is the most attractive point of the platform for you to choose it? So thank you, Dr. Zhou.
Okay, I think the ELISA method is has 3 very attractive points. One, I think, is the ultra high sensitivity. Actually, from our experience for very many different assays, we can, a lot of time we can go to sub-femtomolar sensitivity that consider very high sensitivity. And also because we use 2 kinds of probes, one capture probe and one detector probe. So the specificity would be even better. And another very key important point is that we typically have 5, at least, sometimes maybe more than 5 order magnitude of linear dynamic range. And also and another attractive point is we typically just use a very short analysis time is enough to detect it. And additionally, I think this kind of protocol is very suitable for the using it as a point of care detection. Yeah, I think that's the what I think is the most attractive feature of the ELISA.
Wow, thank you. Thank you for Dr. Zhou. And the next one is for Tony. So Tony, the question is, which biomarkers or applications that INB so far, Instant Biosensors so far have now? And does the platform have any limitation, such as molecular weight or some buffer, etc.? So Tony, please. Thank you.
Thanks for all the questions. So before I answer that, because our company would like to focus on the brain biomarker, which is that neuron size part. So we welcome that we do have the deeply cooperation, for example, and also just like a GFAP, that's one of, and the other is what we develop is NFL. And also the next will be the, we call this the tau, which is the phosphorylated tau antibody. So that will be our three of the major focus of a commercial kit will come in this year. So the based on this, we because the first we will also do the like analysis. The analysis, for example, like NFL, how we develop, we use like 10 different brand and resources. We just do the deeply analysis to find out the best of the combination of the ability and also the atom binding. So so we before we do the detection, we already have a very deep understanding of how the performance of these our brain biomarkers. That's the first step. And also the limitation at this part is if for the analysis, if we want to do the ultra sensitive part, in our mind actually is to the antibody performance, because we are to compare is the ultra sensitivity these level. So the antibody quality should be good, and also the size of the antibody around is up to 5 kilodaltons. That's what we do. Although we run like aptamer or DNA protein is below 5 kilodalton, but we for the ultra sensitivity part, we will highly suggest use the biomarkers up to 5 kilodaltons. Thank you very much. Thank you.
Okay, thank you, Tony, for your answer, and thank you for Dr. Zhou to accept our invitation to join this webinar. And so thank you for all participate to join this webinar today. And we will please keep in touch and connect us directly by email or just text us through the LinkedIn. Yeah, anyway, so welcome to connect us if you have any further ideas or you want to know more about FOPP technology or FOPP platform. So thank you for everybody to join this webinar today. Have a nice day. Have a great day. Thank you very much.
Thank you very much. Thank you.
Thank you. Bye.